Thank you very much. I would like to start with thanking the organization for this invitation. So when Dr. Naif approached me about choosing a topic in CML, I've always wanted to take all the major trials in upfront CML chronic phase and try to compare them. So I thought this will be a good chance for me to do that exercise. So basically, what I'm trying to do today is discussing the major clinical trials in chronic phase CML, trying to bring them all together in one table. Of course, when I thought about doing that, it will be really extremely hard to show one large table with all the different outcomes. But I thought I will break it down into a series of tables, trying to compare those trials. We always hear lectures about CML chronic phase trials, each trial at a time. But I thought if we look at the trials at once, it will give us a better visualization of the outcomes, if it makes sense. So basically what I will try to cover today is the major clinical trials that led to approval of the 5 drugs in the 4 drugs in CML. But I'm not going to cover the ponatinib and radotinib, mainly because the radotinib, based on the RERISE study, was only approved in the Korea. It's usually not used outside Korea. But for the 4 major drugs that we know, based on the IRIS study for imatinib, dasatinib, the DESAGEN trial, nilotinib, which is the Enistin-D, and bosotinib. Bosotinib, there were 2 major clinical trials, the Billat trial and the B4 trial. So the initial publications of these trials, as we can see, the first trial was for imatinib, which is the famous IRIS trial. This was published initially in New England Journal of Medicine in 2003. There was another published update in New England Journal of Medicine in 2017. Of course, there are other updates presented in some local meetings or international meetings, but I only focused on the published manuscripts because they went through the peer review. DESAGEN was initially published in New England Journal of Medicine in 2010. This is the dasatinib trial, but also updated later on in a publication in JCO in 2016. For nilotinib, it was again published in New England Journal of Medicine, actually the same time when dasatinib was published, the same issue in fact, and then was updated later on in the leukemia in 2016. The 2 trials for bosotinib, the Billat trial and B4 trial, Billat was initially published in JCO in 2012, and there was an update 2 years later in the American Journal of Hematology. The most recent bosotinib was the one that was published recently in JCO, and that led to approval of bosotinib in frontline. So if we look at the IRIS study, it included 1,106 patients with chronic phase CML. Those were randomized to either receive imatinib, 553 patients. The same number of patients were randomized to the other arm, which is the interferon plus cytarabine. I think one important message from this trial is that a huge number of patients from the interferon arm actually crossed over to imatinib. So 363 out of 553. So the majority of patients actually crossed over to imatinib. And another point here is a large number of patients, so 111 out of 553 in the imatinib arm actually had data censored due to unknown survival of those patients at 10 years. So basically, there is this very significant number of patients who lost to follow-up, and basically we don't know what happened to them. When we look at the DESAGEN trial, so 547 patients were randomized to either receive dasatinib versus imatinib. In STEND, there were 3 arms of this study. So the intervention group had 2 doses of nilotinib, either 300 milligram BID or 400 milligram BID, compared to the standard dose of imatinib, which is 400 milligram. So about 280 patients in each arm of the study. When we look at the Billat trial, so here we have 581 patients randomized to either receive the standard dose of imatinib versus the bosutinib, about 250 patients in each arm. Before trial, were randomized in a 1-to-1 fashion to receive either the intervention group, which is the 400 milligram of bosutinib once daily, 268 patients, versus the comparison, which was the imatinib in 268. So if we look at the number of patients in the studies, we can tell that imatinib had the highest number of patients, so 1,106. The second largest study is the NST-ND, so 846 patients. The rest of the trials were more or less the same, around 500 plus patients. When we look at age, so these studies included patients who are adults, aging 18 up to 85 or so. The only exception maybe was the Billat trial that included patients up to the age of 91, which is actually impressive. But if you look at the median age of these trials, it's more or less around 50. But this is actually not the case in the real practice. I think Dr. Absi already showed in the CLL trials, patients in the real practice are 10 years younger compared to the patients in the clinical trial. And it's exactly the same thing happening in CML. So these patients in the clinical trial, the median age is 50, but in real life, it's actually 10 years older. So the median age from the cancer registries or SEERS, for example, data and the real patients that we see is around 60. So definitely patients included in the clinical trial are younger compared to the population that we're seeing. When we look at the primary endpoint, you can see that the IRIS study had a survival endpoint, which is event-free survival. In the latest publication, they changed the wording of the primary endpoint to including event-free survival as a primary endpoint, and the long-term primary endpoint is overall survival, which I don't completely understand why they did that in the latest publication. But I think event-free survival is the major endpoint. One disadvantage of using event-free survival or progression-free survival is that these are composite endpoints. They include progression, they include relapse, and they include death. I think maybe some of you agree or disagree with me, but I think having overall survival as one endpoint is better than having 2 or 3 surrogate endpoints together in one. In any way, we use event-free survival and progression-free survival in many diseases, especially those diseases that are chronic. And to me, it's more robust comparing to the other endpoints, although we know that some of the endpoints like complete cytogenetic response or MMR could be translated into survival benefit, whether it's EFS or PFS or overall survival. But of course, when you look at the design of the clinical trials, if you put overall survival or event-free survival as the primary endpoint, your whole calculation will differ. And I'm sure the people who know the statistics will understand this, because the power of the study, the calculation of the sample size is actually quite different if you put event-free survival compared to cytogenetics response or molecular response. And that maybe partially explains why the patients in the IRIS study, the sample size was much larger. Why? Because we wanted to show some survival benefit. But if you need to show some survival benefit, you need to have typically higher sample size. If you look at the other endpoints, the primary endpoints in the other studies, so Billat and Desajan used complete cytogenetic response at 1 year. NST-ND used the major molecular response, defined as less than 0.1% at 12 months. and the before used the same thing, major molecular response at 12 months. Again, these are surrogate endpoints. Yes, they are important, but they're not as important as the survival endpoints. When you look at the key secondary endpoints, each study had a long list of secondary endpoints. So the key ones here for IRIS, for example, the response rate, time to response, disease progression, safety, and side effect profile. The SAGEN used the major key endpoint was the major molecular response. NSTND used the durable major molecular response by 2 years. So their primary endpoint was major molecular response at 1 year. Durable major molecular response at 2 years was a secondary endpoint. A complete cytogenetic response at 1 year and progression to advanced CML, accelerated or blast phase. If you look at the Billat trial, they had a much longer list of secondary endpoints. Now, what is wrong with having a long list of secondary endpoints? Well, we know that the p-value of 0.05 means that there is 1 in 20 risk of having significant value just by chance, which meaning if you have 20 secondary endpoints, there is a chance that one of them will be significant just by chance. So I don't think it is really desirable to have too many secondary endpoints. Basically, it's better to have less secondary endpoints so that the p-value becomes more believable. Before trial used complete cytogenetic response at 1 year, major molecular response by 18 months, duration of complete cytogenetic response and major molecular response, and also event-free survival and overall survival. How about risk scores? So all of the studies used the SoCal score except the SAGEN, where they used the Hasford score. In terms of the sponsoring role of the companies, I think it's still one important thing that we look at when we criticize the articles is to see how much influence was from the drug companies on the design of the trials. This doesn't mean directly that the drug companies will change the result or things like that, but at least for us to know that the drug companies were involved in this and or that. So, for example, the IRIS, the Novartis participated in the design of the trial and the analysis. The Satinib, BMS, were involved in data collection, as well as in analysis, interpretation of the data, as well as writing the initial manuscript. In the NSTND, so the company were involved in writing the first manuscript by actually a medical writer that was employed by the company. There wasn't anything mentioned clearly on the Billat or before trial that I could find. When you look at the median follow-up, there are two numbers there. The first number was the number that was published in the initial publication, and the last one was the most updated one. So imatinib, the median follow-up initially was 19 months. Now we have much longer data, more than almost 11 years or more. The Satinib, initially the duration of treatment was mentioned to be 1 year during the first publication. Now we have data up to more than 5 years. Similarly with nilotinib, as I mentioned, they were published at the same time. For bosutinib, for Billat trial, the duration of treatment during the first the publication was just over 1 year, but the last update was over 2.5 years. The before trial was just released, so we don't have much more update, but the median duration of treatment was 14 months. Now this is, I think, is an important slide, is the percentage of patients who completed treatment in these trials. So now keep in mind these are randomized clinical trials. So basically in real life situation, probably the numbers are higher than this. So 48% at 10 years completed imatinib. So over half of the patients who were on imatinib actually did not complete the trial, either because of a side effect, or maybe they switched to a different drug, or maybe they died, whatever the reason is. More than half of the patients did not complete the treatment. When it comes to the second generation TKIs, so the one in bold here is the numbers at the latest publication. So still, yes, it is better than imatinib, the number of patients who completed the treatment, but it's not as high as we think. 60% or so at 5 years of dasatinib, maybe slightly less with nilotinib, remained on the treatment. So still there is a really big number of patients who did not complete the treatment at 5 years of both dasatinib and nilotinib. We can see similar number, or a trend at least, in the bosutinib, but these data are still quite young and it's not mature enough. I would assume that if we follow these patients with bosutinib at 5 years, we will have similar numbers to the NSTND and the SAGA. How about the progression to accelerated phase or blast phase, or also called advanced CML? So in the IRIS study, so compared to interferon, so imatinib was associated with less chance of progression to CML, so 7.9 compared to 12.8% at 10 years. When we look at dasatinib as compared to imatinib, 5% in the imatinib compared to 5% in dasatinib compared to 7% in imatinib progressed to advanced stage CML at 5 years. Only less than 1%, so 0.7% of patients on nilotinib progressed to advanced stage CML compared to about 5% with imatinib. And in fact, in the NSTND trial, there was no single patients actually, while taking nilotinib, progressed to advanced CML. So the 0.7% were those patients who had to stop nilotinib. So they were on the arm of nilotinib, but did not progress while taking the drug. So they stopped it because of whatever reason, then they progressed. But while taking nilotinib, there was no patients actually progressed to, which is impressive. Billat, we can see a similar trend, so 2% versus 4%, and before trial, more or less the same. So risk of progression to advanced CML was higher with imatinib compared to the second generation TKIs. But you can tell from all these numbers that the absolute difference is not really big. So 2 versus 4% is not really that high. It is significant, it is statistically significant, but the absolute difference is not that high. So in terms of event-free survival, the only trial that I found that they reported this was the IRIS. This is very mature data. As I said, more than 10 years. Event-free survival is still quite significant compared to the comparison arm. So almost 80% are still alive at 10 years, despite all those lost to follow-up patients. When you look at the overall survival, still at 10 years, 83% of patients on imatinib were alive. We can see that with dasatinib, 91 to 90% the difference between dasatinib and nilotinib. This was not statistically significant. Similarly, between the nilotinib versus imatinib, 94% versus 92% at 5 years. Again, this was not statistically significant. Similarly, in the bosutinib trials, although overall survival was not different between imatinib and bosutinib. So there was basically no overall survival benefit. When we look at the death at the end of the trial, so 16% of patients while on the imatinib arm died at 10 years. Dasatinib, 10% taking dasatinib versus those who took imatinib at 5 years, so exactly the same, versus 6 versus 7% in the nilotinib, so no big differences at 5 years. And in the Billat trial, 3 patients died while on imatinib, on bosutinib versus 8 while on imatinib. Similar trend in before trial. How about complete cytogenetic response? More or less, we can see that the numbers are close to each other, around 80% while on the newer TKIs, 80 to 70%, compared to about 60% in the initial publication versus 82%. So there is a trend for imatinib to reach deeper remission while the study is ongoing. A major molecular response is seen in higher frequencies in the second generation TKI compared to the first generation TKI. So for example, the dasatinib, 76% with dasatinib compared to 64% at 5 years with imatinib. 77% with nilotinib achieved MMR compared to 60 at 5 years. If you look at the IRIS study, 34% only of all patients achieved this. However, because of that large amount of patients who lost to follow-up, only 204 patients were evaluated, or they could be evaluated for MMR. Out of those 204 patients who could be evaluated, 93% were in MMR. So this is, I think, it's unanswered question for imatinib, but if we see the comparison between imatinib and the second generation, it will be around 64%, at least in 5 years. We don't know that in 10 years. This is one of the important endpoints that we look for. Why? Because it helps us decide on the discontinuation or treatment-free remission, TFR, which will be discussed in the next lecture. Basically, what is the percentage of patients who might achieve MR 4.5 in imatinib compared to second generation TKI? So for imatinib, this number was around 23%. But again, keep in mind only 204 patients could be evaluated out of 553. So we don't actually know the exact number. It could be higher than 23%. But when you look at the number for imatinib in dasatinib as well as NSTND, it is about 33%. So about 30% or so of patients on imatinib will achieve 4.5, as compared to 40 to 50% on the TKIs will achieve 4.5%. I think this is an important slide. I think we need to keep this in mind while we're comparing these first versus second generation TKI. The difference is significant, but it's not that huge. So just to summarize, overall survival is not different between first and second generation TKIs, as we know. Imatinib data was based on strong endpoints, based on survival. We had larger sample size and the mature data. And it is becoming very hard to show survival differences in between the TKIs themselves. Why? Because imatinib was so effective to begin with. It showed improvement in survival, and now we're comparing the newer drugs with a drug that is very effective. And that also explains why the primary endpoints were different in those studies, because it's really hard to show survival benefit now. Keep in mind that more than 50% of patients will not remain on imatinib at 10 years. So most of them will be switched to another drug, versus 39 to 41%, which is still a really significant number of second TKIs will not be on the drug at 5 years. If you look at progression to the advanced CML, nilotinib had the lowest rate of that compared to the other drugs. When you look at the MR 4.5, which is relevant to TFR, as we mentioned, with imatinib, it is ranging between 20 to 30% or so, as compared to 40 to 54% with the second generation TKI. But one important factor that I always try to emphasize, and I think it's really important for us to know, that the TFR can only be achieved in about maybe 10% of patients, and probably less than this. Where does this number come from? If you look at MR 4.5 of 50%, for example, in second generation TKI, So all comers treated with second generation, only 50% will achieve MR 4.5, so half of them. And 80% of that half will be eligible to stop. So they have to be in deep remission for at least 2 years, et cetera, et cetera. So if you apply that criteria, only 80% of them will be eligible to stop or eligible for TFR. So 80% of the 50%. Ultimately, you'll end up having 10 to 15%, and this is based on randomized clinical trials, be able to stop those drugs. But again, keep in mind, 10 to 15% is in randomized clinical trials. So what is this number in real life? Nobody knows. This will be a really good study to answer. Real life discontinuation rate in CML. I think it will be lower than this. So at the end, TFR, yes, it is important. I still look at it. I discuss it with my patients, especially young patients. But let's not give it a big hope, because only 10% of our patients will achieve that. So thank you very much.
So thank you, Dr. Musa, for an excellent presentation and summary. The floor is open for any questions. No question.
Musa?
Thank you, Dr. Musa. You highlighted very important challenges that we have in terms of endpoint selection. And in fact, that we keep lowering our standard in terms of the surrogate endpoint. So for other diseases, we started accepting MRD as a surrogate endpoint for drug approvals, not just for as endpoints for trials. But the challenge that we have also is how to move forward. Like, yes, everyone wants to show an overall survival advantage, or let's say a meaningful endpoint in terms of that patients and physicians will be happy about. But how can we, is there any reasonable way that we can achieve this goal without necessarily having to wait for 15 years or 20 years before we can answer certain questions?
Mm-hmm. Well, I think it is a very important and tough question to answer. It's not really easy. And that is part of the reasons why they're not including these outcomes from the beginning. I think one thing to think about is when you want to show some survival benefit, let's say, for example, if you want to show survival benefit of imatinib versus nilotinib, you have to have very large sample size in order for you to show that small difference. So in order to show that, first of all, the drug companies will not be interested in answering that questions. Why? Because imatinib now is generic, right? So, and we show that many of the companies are involved in the design of the trials. Let's just be honest. So nobody will be interested in running that trial of comparing imatinib with that. But let's just assume that there is a drug company or investigator-based RCT where you want to compare imatinib with a second generation TKI. Let's just assume that it will happen. You will need to have a very large sample size. And in order for you to do that, you have to collaborate with so many centers so that you include patients with that. So I think technicality, I completely agree with you. It is difficult, it's not easy. I think we'll not be seeing a lot of these trials where we have really important outcomes. We will still see a lot of surrogate endpoints, a lot of less important endpoints. That is the reality. But the thing that I wanted to say is that we as physicians, we have to recognize that reality. Not to be, not to take the result for granted. We always have to question the results. We have to think about what outcomes are used in the studies and why do we use it. What is the benefit for these studies and the patients that you see in the clinic? Because when the patient comes to you in the clinic, he wants to know why this drug is better than this. Is it going to prolong my survival? Or is it going to decrease the risk of relapse? Or is it going to deepen the remission? This is what the patients ultimately want to ask you about. But a very important question. Thank you.
Yeah, I agree with you, Musa. I think it's very important the first visit of the patient to know what's really what the patient wants. Is it like if you're seeing a young female patient or you're seeing an old male patient, it make a difference. So if the patients clearly prefer to have the medication stopped later in life, you would try to give them the best chance of that by what you kind of collect and understand from the clinical trials. And if it's something else, then you can try something else that is less expensive, etc. Just a comment.
Yeah, I agree with you.
Dr. Bayroti?
This is a great effort, yeah, Musa. It's really great effort, and you know, in institutions, they do have the chemotherapy therapeutic subcommittee, and then they have the formulary committee, or the yeah, it's called formulary committee. And in these committees and subcommittees, they go what you did. They go really, and they add additional evaluation, which is the cost, and what's the impact of the cost. But to be honest, we don't tell the patient all this information for two reasons. First, I don't know if the patient will understand surrogate markers. Second, I'm not sure that even if the speaker will understand what he's talking about. So it's really difficult situation. So what we will tell the patient, we will go to guidelines, and that's what's happening. So in the guidelines, like NCCN guidelines, which is a relieving variable, that this is approved in the guideline, NCCN guideline. That's a relieving, because the exercise you did is really reviewed. So other countries like UK, they have another meaningful evaluation, which is the NICE. And NICE is evidence, but they put the cost. And it's another caveat. In Saudi Arabia, I don't think we have the, it's institution-based, but countrywide. I hope with this, now the Majlis al-Daman al-Awram, they can really start to look and establish guidelines for the country, look at this, and when the drugs approval, and the Saudi FDA, they really accept whatever European or American, but to accept these markers as a positive or negative, and that's why you'll see differences. Some drugs got approved in USA, not approved in Europe, and some, and because of these differences that you mentioned. So it's very tricky, and at the end of the day, we should make it simple, practical, and I don't know when will be the day we will use artificial intelligence to guide or decide on our behalf.
Yes, I agree. It's a difficult balance between trying to improve the individual patient's outcome versus policy making, you know, with population-based decisions. So it's not easy. We have to take into account the cost of the medications and cost effectiveness and all. I agree.
Thank you, Dr. Zahrani.
Thank you.
For this nice talk. And we're going to move to the next talk, which is about the risk adopted.